Our second lecture for Unit 2 focuses on biological catalysts, or enzymes. A catalyst is a substance that speeds up the rate of a chemical reaction, but is not itself changed by the reaction. Enzymes act as biological catalysts. They occur inside cells or are secreted by cells, and the suffix ase indicates an enzyme. Enzymes are generally proteins. The proteins are, again, as you remember, are long molecules that are folded into a specific shape. You see here cartoons of different shapes of many different enzymes in the cell. Notice again the suffix ase for most enzymes can tell you that it is indeed an enzyme. Occasionally, though, the name is slightly different, as in the case of the digestive enzymes pepsin and trypsin. While these are cartoons on the top of the slide, I actually imported what the structures are for each of these particular enzymes beneath the name. Again, it'll show you or help you remember some of your protein lectures dealing with the different types of structure. You should be familiar with here. You see some of these secondary structures, these alpha helices here, as well as these more flat regions. You see the beta sheets exampled here in purple. In the example of catalase, you also see that there's quaternary structure. You see 4 different subunits coming together to form the enzyme catalase. In order for the enzyme to function properly, it needs to be folded properly, and so the amino acid sequence of the particular protein is often critical for it to ultimately perform the necessary function, or I should say to fold properly and then perform the necessary function. The part of an enzyme, there's a couple of different regions of enzymes that we'll cover that are important. The part of an enzyme where the reaction occurs is known as the active site. This is one region that you should certainly be familiar with. The molecule the enzyme acts on then is known as the substrate. So the substrate would bind to the active site, and then ultimately the molecule the enzyme produces is known as the product. So the sequence goes substrate binds to active site. Ultimately then the enzyme acts on the substrate to produce a particular product. A unique feature about enzymes is that they can be reused or recycled in the cell. And so once the substrate has bound and the reaction has occurred, the product will dissociate from the enzyme, and the enzyme can then be or is available to bind to another substrate and ultimately create another reaction. And so here you see enzymes being reused over and over again, substrates binding, reaction occurring, product comes off. Each enzyme is specific to a particular substrate molecule or molecules, depending on what type of reaction the enzyme is ultimately working on. The lock and key hypothesis states that the active site specifically matches the shape of the substrate molecule. And so then that's what gives you that sort of lock and key. Each enzyme has a conformation or a shape that makes it particular for the substrates that it'll bind to. Now, the lock and key hypothesis in these cartoons is a little bit simplistic. Often what we will find is that the enzymes have a conformation or a shape that allows it to bind to the substrate. The proteins, though, are not perfectly static or stuck, if you will, molecules. They're constantly changing slightly in shape, although the overall global structure or architecture of the enzyme molecule is specific for its particular substrates. The rate of an enzyme is controlled in essence by temperature. At very low temperatures, enzyme-controlled reactions go very slowly because the molecules have very low kinetic energy. But in contrast, when you increase the temperature, the reaction also increases. As the molecules have more kinetic energy, they're binding quicker to the enzyme. The enzyme can perform the particular reaction that it needs to produce a product, and then the product dissociates. But I will also say that there is a maximum temperature at which the reaction will occur optimally, and then beyond that maximum or optimal temperature, you actually have the reverse. So it only occurs about optimum temperatures, usually around 40 degrees Celsius. And again, as I mentioned, the temperature at which the rate of the reaction is the fastest is known as the optimum temperature. When you get higher than that optimum temperature, what happens is the proteins start to denature, or in essence unravel in their structure or architecture. So the enzyme begins to change shape, and the active site no longer then matches the shape of the substrate molecule. And so as you change that active site, you no longer have the ability of the substrate to interact with the enzyme. And so what you see here is substrate not capable of binding to the enzyme any longer because you have lost that functional active site. Rates of enzyme reactions can be measured by recording the time for a substrate to disappear or for the time that it takes for a certain product to appear, depending on what you're particularly measuring. So in an example here, you have protein is white. When it's acted on by the enzyme trypsin, it's broken down into individual polypeptides, which are then clear in a particular test tube. And so you can measure this rate of reaction. And you can change different things then as you're performing these experiments to learn more about how an enzyme would work. And some of the variables, as you see here, that you can change is you can change the volume and the concentration of the substrate. You can change the volume and the concentration of the enzyme. Not only does temperature affect the rate of reaction, but pH can also affect the rate of reaction because, again, it acts or changes the structure of the enzyme proteins. And then temperature, as we already mentioned, can affect reaction rate. And so here we see if we plot on the x-axis temperature, and you see an increase in temperature versus the rate of reaction, you see the reaction increases until it reaches that optimum temperature. And then as the protein begins to denature or fall apart greater than 40 degrees Celsius, the reaction starts to slow down because the enzyme is essentially no longer functional. So here you see molecules gaining kinetic energy in the beginning of the curve. You've reached the optimal temperature, followed by an enzyme denaturing. Now, as I had mentioned, not only does temperature affect the reaction, but pH does. And so within an optimal neutral buffer pH, you see the enzymes working optimally. Again, it depends on the enzyme, though. For most physiological reactions, when we deal with an optimum temperature, or pH range, I should say, we're dealing with this 5 to 9 pH range. What you see for pepsin, this is an enzyme that's working in the stomach, where actually, because of the environment there, the enzyme has it has a structure that allows it to work or actually be optimal even in a very low pH environment. And so pepsin is a little bit of an outlier, being that it's a digestive enzyme. There are other digestive enzymes that are also optimal at very low pHs, I should say. But most of the physiological enzymes in our body are working at a much more neutral pH range, somewhere around 6 to 7. Now, you come in contact with enzymes every day, not only in your body, and we'll learn about several of them as we go through these units, but they're also important in sort of our everyday life. And so an example of enzymes in motion is we often see or have enzymes included in our laundry detergents. An example of that, if you see an ASE somewhere on your laundry detergent box, proteases. These are break down the colored insoluble proteins that tend to cause stains and allow you to wash your clothes at lower temperatures. If I back up for a second, you also see here another enzyme on this slide. It's a lipase or a lipase. Some people will say either one is fine, and you can probably already guess what this enzyme acts on. It acts on the lipids to dissolve the grease or dirt, in essence, any of the fats that are staining your clothes. Enzymes are also often used in the food industry. You see pectinase breaks down substances in the apple cell walls, allows you to get more juice from the apples. Lactase breaks down the lactose in milk, makes milk drinkable for lactose intolerant people. Let's take a second to review how enzymes work.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
And so this was just a quick introduction into enzymes, what they are, what they do. Enzymes and metabolism go hand in hand. We'll see a number of different enzymes coming up as we go through the next lecture when we talk about ATP production and the Krebs cycle.